Fatty acid synthesis. The process of fatty acid synthesis was studied by Linen, who got Nobel Prize for this in the year 1964. The pathway is referred to as linen spiral. The dietary carbohydrates and amino acid, when consumed in excess, can be converted to fatty acids and stored as triacyl glycerol. It is, remember, it is not a reversal of fatty acid oxidation, which I can show you here for better understanding with the following table, which shows the comparison between these two metabolic pathways, and the synthesis of fatty acids and their oxidation are two distinct and independent pathways. And the site for the fatty acid synthesis is synthesis of fatty acids occur predominantly in liver, lactating mammary glands, adipose tissue, kidneys, brain and lungs. The enzyme machinery for fatty acid production is located in the cytosomal fraction of the cell, and the requirements are acetyl-CoA and NADPH. The acetyl-CoA is a source of carbon atoms, while NADPH provides the reducing equivalents, and ATP supplies energy for fatty acid formation. The major fatty acid synthesized is palmitate, which is a 16 carbon saturated fatty acid, which is further elongated and desaturated to form other form of fatty acids. And the sources of acetyl-CoA and NADPH are: acetyl-CoA is produced in mitochondria by the oxidation of pyruvate and fatty acid and the degradation of carbon skeleton of certain amino acid and also from the ketone bodies. And the sources of NADPH, which comes from the HMP shunt and the malic enzyme. So here the malic enzyme, how it acts as a source. Here the malic enzyme converts malate to pyruvate. The NADPH and CO2 are generated in this reaction. Both of them are utilized for fatty acid synthesis. Now begins with the fatty acid synthesis. Let us try to understand the fatty acid synthesis in three phases. In the first phase, transport of acetyl-CoA. Because here the synthesis should occur in the cytosol or cytoplasm. Now the acetyl-CoA is present in the mitochondria, and this is impermeable. The inner mitochondrial membrane is impermeable to acetyl-CoA. So the acetyl-CoA should come out as a citrate. So how it comes out? Let us try to understand here. The acetyl-CoA, which is generated in the mitochondria by the oxidation of fatty acid or oxidation of pyruvate, then this acetyl-CoA should get condensed with oxaloacetate in the mitochondria to form citrate. The citrate, which is freely transported across the membrane. So citrate is freely transported to the cytosol, where it is cleaved by citrate lyase to liberate oxaloacetate and acetyl-CoA, which then involves in the fatty acid synthesis. Now begins with the phase 2, formation of malonyl-CoA. Here the acetyl-CoA is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. This is an ATP dependent reaction and requires biotin for CO2 fixation. Here the A, B, C is required, that is ATP, biotin, carbon dioxide, and also here the acetyl-CoA carboxylase is a rate limiting enzyme in the fatty acid synthesis, and which is well regulated, and it is a rate limiting enzyme. So now of the 16 carbon palmitate, 2 carbons are added in the form of acetyl-CoA at the omega end, and the 14 carbon come from malonyl-CoA. Phase 3, the fatty acid synthesis proper, which is the fatty acid synthase, the major enzyme for fatty acid synthesis, which contains 7 different enzymes. So let us try to understand this fatty acid synthase complex with the diagrammatic representation here. The two subunits lie in anti-parallel head to tail orientation. The SH group of phosphopantethine of one subunit, which is attached to the ACP, is in close proximity to the SH of cysteine residue of the enzyme ketoacyl synthase of other subunit. Here let's see here, the ACP has the 4-phosphopantethine, which is having SH group which is close proximity with the SH group of cysteine residue of the ketoacyl synthase. So now it is a multifunctional enzyme, also known as multi-enzyme complex. It has activity of 7 enzyme and 1 protein, that is acyl carrier protein, and it is a dimer composed of 2 monomers, and dimer is only functionally active. The multi-enzyme polypeptide complex is coded by a single gene. Thus, there is a good coordination in synthesis of all enzymes of fatty acid synthase complex. All these enzymes are in equimolar concentration. They are well regulated and does not interfere with the other metabolites. So here, which is having a condensing unit and reduction unit and also the releasing unit, and it is functionally this is the functional division and the subunit division. Now the 7 enzymes are, these are the 7 enzymes which you can able to remember with this mnemonic. And let us begin with the reactions of fatty acid synthase complex. Now the first reaction is the 2 carbon fragments is transferred to the ACP of the 2 carbon atoms transferred to the ACP of fatty acid synthase, catalyzed by the enzyme acetyl transacylase, which can also be called, we can call as acetyl CoA ACP transacylase. The acetyl unit then transferred to the cysteine residue of the enzyme. Thus this side fall vacant. So now it forms acetyl S enzyme. This acetyl S enzyme, which is having the acetyl group at cysteine residue, now this is vacant. ACP is having vacant at SH group. Now the enzyme malonyl CoA ACP transacylase or malonyl transacylase transfer the malonate or malonyl CoA, and this malonyl moiety loses carbon dioxide, which was added by the acetyl CoA carboxylase in the before previous step. Thus CO2 is never incorporated into the fatty acid carbon chain, and the decarboxylation is accompanied by loss of free energy, which allows the reaction to proceed forward. This reaction is catalyzed by beta keto acyl synthase. Now it forms beta keto acyl ACP. Now let's see this beta keto acyl, which reduces by the beta keto acyl reductase, reduces keto acyl group to hydroxyl group. The reducing equivalents are supplied by NADPH, and the beta hydroxy acyl ACP undergoes dehydration. A molecule of water is eliminated and a double bond is introduced between alpha and beta carbons. Now this forms trans-enoyl ACP or trans-enoyl ACP. Let's see what happens to this. The second NADPH dependent reduction catalyzed by enoyl reductase occurs to produce acyl ACP. The 4 carbon unit attached to the ACP is butyryl group. The carbon attached to ACP is transferred to cysteine residue, and the reaction 2 to 6 are repeated 6 more times. What is this 2 to 6 reactions? Let us go back to the second to sixth reaction, which we have seen already all this. This is the second reaction, like this acetyl S enzyme combines with malonyl CoA. From this step to the sixth here, the trans delta to enoyl ACP till here repeats 6 more times. So each time the fatty acid chain is lengthened by 2 carbon units obtained from the malonyl CoA. At the end of 7 cycles, fatty acid synthesis is complete, and 16 carbon fully saturated fatty acid, namely palmitate bound to ACP, is produced. So now this palmitate should be released. How it should be released? It from the ACP. Let's try to understand here. The enzyme palmityl thioesterase or thioesterase separates palmitate from fatty acid synthase. This completes the synthesis of palmitate. Let's begin the summarizes. The net reaction of synthesis of fatty acid may be summarized as one which are utilized are 1 acetyl CoA plus 7 malonyl CoA plus 14 NADPH molecules, which forms 1 palmitate, 7 carbon dioxide plus 14 NADP plus 8 coenzyme A plus 6 H2O will be released into the reaction. So now it should regulation. Coming to the regulation, how it is regulated? The acetyl CoA carboxylase, which is a rate limiting enzyme, and the regulation enzyme is regulated by two mechanisms, that is polymerization, phosphorylation. Polymerization, acetyl CoA carboxylase is active as a polymer. So polymerization is done by citrate, and the phosphorylation, which is inactive, AMP activated kinase. Though now this enzyme is committed step in fatty acid synthesis. So acetyl CoA carboxylase exists as an inactive protomer, monomer, or the active polymer. So the citrate promotes polymer formation, hence increase fatty acid synthesis. On the other hand, and the palmityl CoA and the malonyl CoA causes depolymerization of the enzyme and therefore inhibits fatty acid synthesis. And let's begin with the regulation with the hormones. And hormones regulate acetyl CoA carboxylase by a separate mechanism: phosphorylation, which is inactive form, and dephosphorylation, active form of the enzyme. Glucagon, epinephrine, norepinephrine inactivate the enzyme by CAMP dependent phosphorylation, and insulin on the other hand dephosphorylates and activates the enzymes. Thus insulin promotes fatty acid synthesis while glucagon inhibits fatty acid synthesis.